Olá. Bom dia novamente a todos. Peço, por favor, para quem está de pé, para que se acomode. Para darmos início à nossa última sessão. Bom, meu nome é Aline Lorenzon, eu sou gerente científica do Grupo Huntington, faço parte da comissão científica e com muito prazer, então, vou conduzir aqui o nosso segundo painel do dia, onde continuaremos a falar um pouco mais sobre o futuro das técnicas de reprodução assistida. It is my great pleasure and honor to introduce you our first speaker, Professor Graham Burton. Dr. Burton is the Mary Marshall and Arthur Walter Professor Emeritus of Physiology of Reproduction and the founding director of the Centre for Trophoblast Research at the University of Cambridge, UK. His lecture is titled The Unrevealed Mechanism of Embryo Implantation. Welcome back again, Dr. Burton.
Thank you, Aline, for that introduction and the opportunity to come to this wonderful city. As a non-clinician, I have learned an awful lot over the last 2 days. So the title, The Unrevealed Mechanisms of Embryo Implantation, I mean, in the human, I think this is still, we're very ignorant about the basic processes of implantation. Why? Because it's largely a hidden event, and there are huge species differences in implantation, placentation, between species. So really there's no good animal model for the human. You'd have to go to the great apes, and that's not really ethically acceptable. The in vitro systems we'll have a look at, but they are limited, but I think there is some potential for the future. So what I'm going to do is just to reveal or to discuss some of the molecular mechanisms that are thought to underpin embryo implantation, because as we heard yesterday in the clinical session, implantation failure is still one of the major blocks to pregnancy success in ART. So just to provide an overview, as we heard again yesterday, implantation requires a receptive endometrium and a competent blastocyst. And there is an extensive crosstalk which is beginning to be elucidated, and we'll look at some of the factors that are involved in that between the endometrium and the blastocyst prior to implantation. And we traditionally recognize 3 phases of implantation: the apposition, the adhesion, and then the actual trophoblast invasion. And we've got apposition, adhesion there. And the molecular mechanisms involved are in fact remarkably similar to those involved in a neutrophil rolling along the endothelium and then extravasating from the vascular system into the tissues. So it requires actually interactions between L-selectin, which is expressed on the surface of the trophoblast, and glycosylated ligands on the endometrial surface. And the glycosylation refers to the way that various sugar groups are added to a protein backbone. So most surface proteins on a cell will be heavily glycosylated, and we'll see the importance of that in a minute. So when I started off in looking at reproduction, certainly the traditional teaching was that the endometrium is in a non-receptive phase for most of the cycle, as you heard of the window of implantation yesterday, and that is because there are a family of mucins, and MUC1, MUC16 are the two transmembrane mucins which are adherent to the surface epithelium of the uterus. And these are very long fibrillar proteins which extend beyond the extent of the receptors on the endometrium for L-selectin. And so for most of the cycle, there is no physical interaction between L-selectin and the receptors here because of the steric hindrance of MUC1. And MUC1 is under the control of progesterone. It is secreted from the glands, and it becomes adherent to the surface epithelium. Now, what you will see in the window of receptivity in domestic species, in the macaque, for example, is that at that time of implantation, there is a wave of reduction of MUC1 in the uterine epithelium, allowing those interactions to take place. And that was thought to be, the answer in the human. But in the human, there is no wave of deglycosylation of MUC1, no evidence that the same sort of mechanisms take place. So what does happen? Well, again, in the absence of an animal model, people have used in vitro models. And here was quite an influential publication at the time where they took uterine epithelial cells, they've stained for MUC1, fluorescent green here, a monolayer of cells, and then they applied blastocysts onto that monolayer. And what they observed was that where the blastocyst was attached, there is a reduction actually in MUC1. This is at the low power image, this is the higher power image, so that cell there corresponds to this cell here, that one there, etc. And when you actually map out the fluorescence intensity across that, if you like, implantation site, you can see that there is a sudden reduction in the extent of MUC1, actually where the blastocyst was attached. So there seems to be some local interaction which is much more subtle, much more discrete in the human than it is in non-human primates and in the mouse. So that's been the sort of understanding for a number of years. The factors that are regulating that MUC1 are not known. But there's a paradox because MUC1 is under progesterone control, and so paradoxically, its expression increases during the secretory phase. So how come you've got increased MUC1 and yet here you've got a reduction at the implantation site? Well, some recent data suggests that we may be able to reconcile these two situations. So a group took two epithelial cell lines. Again, this is all done in vitro, so we must consider all the caveats associated with that. But they used atomic force microscopy to actually measure the binding of the L-selectin on the tip of the microscope to Sial-Lewis X on the epithelial cells. And what they showed was that if you increase the expression of MUC1 and increase its glycosylation, you actually increase the strength of that adhesion. And if you decrease the MUC1, you reduce it. That only applied in that one cell line. It didn't apply in the other cell line. And that what they also found was that MUC1 can carry sialyl Lewis X, this tetrasaccharide here, which is thought to be very important for implantation. It's the same as what is used in the neutrophil rolling mechanism, and in fact the sperm binding to the zona pellucida. So there's this sort of conservation here. So it may be that what is happening is that the MUC1 is increasing in expression, it is increasing its glycosylation, and that there is binding actually of the L-selectin onto MUC1 at the implantation site, and that then there is a local degradation of the MUC1, which allows the L-selectin to actually bind to sialyl Lewis X sitting on the endometrial surface here. So this is a very strong interaction and will attach the blastocyst to the epithelium, and that will be reinforced then by binding of N-glycans on the trophectoderm surface with integrin V beta 3, alpha V beta 3, which is also expressed on the endometrial surface. So that is, I think, trying to reconcile what is happening on the endometrial surface. We also have to think of what's happening on the trophectoderm, and I think we know even less really about that. I mean, it is known from, obviously, that the blastocyst is orientated so that the inner cell mass is opposed to the uterine epithelium at the time of implantation. So this is one of the earliest images of implantation here from the Carnegie collection, and yet there's the probably bilaminar germ disc there attached to the embryonic pole. So what is it about that trophectoderm that is different over the inner cell mass that makes it more receptive to the endometrial epithelium? Is there a difference in the expression of L-selectin? I don't think there's any evidence that that is the case. What we do know is that the fibroblast growth factor receptor 1 is expressed preferentially over the inner cell mass, and the CCR7 receptor again is expressed preferentially over the inner cell mass. So there is evidence for a microheterogeneity within the trophectoderm, but as far as I'm aware, nobody knows what the association between the expression of these receptors and the actual implantation orientation is. But it is, of course, important that the blastocyst does attach in that orientation so that when the umbilical cord develops, it is over the area of the developing placenta, and you don't get a velamentous attachment. So that's the apposition and the adhesion. What about the invasion phase? Well, there are classically 3 different forms of invasion seen within the mammalian order. You've got displacement here where the uterine epithelium is lost. There's the trophectoderm there, and there's an abutment of the trophectoderm to the stroma. There is in the ungulates, in the cow, for example, you see a fusion between the trophectoderm and the uterine epithelium, so you get a mixed materno-fetal epithelium forming. And then there's the intrusion form, which is where the trophoblast actually penetrates between the uterine epithelial cells, and that is the form that will occur in the human. So that has been modeled by taking blastocysts and applying them to the monolayers of endometrial cells, what you will see is that the trophectoderm of the blastocyst that is opposed to the epithelial cells undergoes the syncytial transformation. It becomes a multinucleated syncytiotrophoblast, and it is tongues of that syncytiotrophoblast that penetrate between the epithelial cells. So there's no destruction of the epithelium. They will get junctional complexes forming between the two cell types, and then those the extensions of the syncytiotrophoblast penetrate into the stromal tissue, and that gives you invasion. So what are the mediators and that are potentially influencing this dialogue between the two cell types? Well, there can be soluble hormones and cytokines coming from the blastocyst, but there's a lot of interest at the moment in terms of extracellular vesicles and generally in cell communications. So extracellular vesicles can be of two different types. There are the exosomes, which are about 30 to 150 nanometers in diameter, so these are very small vesicles. And then there are the larger microvesicles, which are 100 to 1,500 nanometers. And they're formed in very different ways between the cells, so that the microvesicles actually bud from the surface of the cell and then are released, whereas the exosomes are formed in a multivesicular body or a late endosome, and then they are released again from the surface of the cell. And these are now known to carry quite a variety of different cargoes. They can contain proteins, bioactive lipids, messenger RNA, and also a wide variety of microRNAs. And so they can have a very profound influence on their target cells. Are there any evidence that these are taken up by the epithelial cells? Well, a group took culture supernatant from blastocysts, which had been fluorescently labeled, and just took that supernatant, applied it to endometrial epithelial cells, and you can see that the cells took up those microvesicles and they became fluorescent themselves. So yes, endometrial cells will take up these exosomes, microvesicles coming from trophoblast. So there's been a sort of profusion of papers, I think, showing potential mediators of this dialogue that are coming from the blastocyst and influencing the uterine epithelium. So we've got secreted factors like HCG, interleukin-1 beta, known to be very important in receptivity, and also a wide variety of microRNAs that are coming from these extracellular vesicles. So a large number of potential mediators. Are they actually important? Are they critical? Very difficult to say at the moment. I mean, I think work on the mouse showed that the only factor that is really essential for implantation is leukemia inhibitory factor, LIF, which is coming from the uterine glands. So if you knock that out, you don't get any implantation. If you knock out some of these other factors, you will get a reduced level of implantation, but you will still get some, so they can't be absolutely essential. So I think there are a number of, the field at the moment is sort of exploding with potential mediators. I'll just show you an example of how one of these, IL-1 beta, is operating, and how that can mediate this. So what is known is that IL-1 beta and LIF itself, IL-1 beta is secreted by the trophectoderm, the decidua and the maternal immune cells, whereas LIF is coming from the actual glands. And what they do is to actually increase the expression of Sialyl-Lewis X on the endometrial cells through their actions on these enzymes, the fucosyl transferases. So here is IL-1 beta binding to its receptor. That then signals to the nucleus of the epithelial cells to increase this enzyme FUT3. The protein is located in the endoplasmic reticulum, where you get the glycosylation and the Golgi body, and then that increases the expression of Sialyl-Lewis X on the epithelial surface, which then binds to that L-selectin. So this is Sialyl-Lewis X here. This is a tetrasaccharide there, and you can see that it has a fucose added to the N-acetylglycosamine here. So that enzyme, the FUT3, is actually mediating this fucosylation, and that is generating the Sialyl-Lewis. So what they did was to take some blastocysts, apply them again to a monolayer of endometrial epithelial cells. In the red bar here, this is the sort of control without any additions. If they added in IL-1 beta, and in the next column along, you see an increase in the percentage of adhesion. On the other hand, in the green bars, if you, the first one is a control again, and then they added in an antibody against Sialyl-Lewis X, and that reduced adhesion, as you would expect. And then the final bars are when they combine the two effects. Again, you got an increase in adhesion with the IL-1 beta and a reduction when you added in the antibody to Sialyl-Lewis X. So I think this is beginning to show you some very nice ways in which this receptivity is actually mediated through factors such as IL-1 beta and LIF. Okay, how can we take this forward? How can we really dissect out some of these mechanisms? Well, human blastocysts have been grown on monolayers of endometrial cells for many years now. And yes, they do show us the interactions and that penetration by syncytia trophoblast, but they're rather limited in their biological, physiological relevance because of lack of paracrine signaling. What about using the organoids that we talked about yesterday? Well, they have the benefit that you have genuine trophoblast and endometrial cells, and they can begin to look at some of those interactions. But again, they lack some of the paracrine signaling coming from decidual cells and possibly maternal immune cells. So what people are beginning to do is to generate assembloids, where you start with the gland organoids and then surround those by stromal cells, and then you get this sort of mixture here. That is the assembloid, the organoid there, the epithelial surface, and then the decidual cells around the outside. And what they've done is then to actually make a little nick in the apex of the dome and to put in a blastocyst there. And you can see, I hope, that you're beginning to get some outgrowth from the blastocyst, again with the polar trophectoderm opposite the inner cell mass into this matrix. Now that looks very encouraging, but of course, in real life, the blastocyst never interacts actually directly with the stromal cells. You need to get an epithelial surface really to look at that interaction. We started to try and bioengineer this by taking that collagen scaffold again. We can apply stromal cells onto that scaffold so that they grow over the collagen. Those stromal cells can be decidualized. This is looking at the change in prolactin in response to estrogen and progesterone. So you can decidualize those, and then we can grow gland epithelial cells over those stromal cells. So at least now you can get an epithelium which is ciliated and the right sort of tissue for interactions with those blastocysts. But there's still a long way to go, obviously, in terms of adding in immune cells and getting a really functional endometrium. But I think, again, this is going to happen within the next few years. So just to summarize, I think there's a very complex two-way dialogue taking place between the trophectoderm and the endometrial epithelium, involving hormones, cytokines, and these extracellular vesicles, leading to a change in receptivity, in the mucins being expressed. Many factors have now been implicated in the implantation. I think their roles are still very uncertain. They may just be bystanders. These are sort of association studies rather than real mechanistic studies, though, as I showed you, we are beginning to see how factors such as LIF and IL-1 beta might influence the system. I think there's mounting evidence that glycosylation plays an absolutely critical role, and that may be influencing recurrent implantation failure or early pregnancy loss, possibly even later complications of pregnancy. And what can affect glycosylation? Well, you need to have the substrates. You need to have the fucose, the glucose available, and that relates to the microbiome again that we heard about yesterday. And of course, glycosylation takes place in the endoplasmic reticulum and Golgi, two organelles that are very susceptible to cell stress. So if you have stress, you may get aberrant glycosylation leading to defective feto-maternal crosstalk. But I think in the next few years, as I say, that bioengineering techniques are going to allow us to dissect out these interactions in much more detail. You may identify biomarkers. We may get targeted therapies in reproductive glycobiology. It's a very complex area, but things are moving now at a fairly rapid pace. So I'm going to leave it there, and thank you for your attention.